Problem 1 states that assume that two pigments, red and blue, mix to give the normal purple color of Petunia petals. Total lack of pigment results in a white petal. Red pigment forms from a yellow intermediate that is normally at a concentration too low to color petals. A third pathway, whose compounds do not contribute pigment to petals, normally does not affect the blue and red pathways. But if one of its intermediates, white 3, should build up in concentration, it can be converted into the yellow intermediate of the red pathway. Blue mixed with yellow gives green. We have to deduce which combinations of true breeding parental genotypes could be crossed to produce F2 progeny in the following ratios. And so evidently we have done a dihybrid crosses, and we are getting modified 9:3:3:1 ratios. So let's start with the first one. This 9:3:3:1 ratio suggests that we have no interaction, or the two genes are acting in two separate pathways. Furthermore, we are getting purple, and that means the two pathways must be the first pathway, which converts white 1 intermediate into blue, and the second pathway that converts white 2 intermediate into yellow, and then from yellow to red. Now, the second pathway has two genes. Is it A, which is mutant here, or is it B, which is mutant here? If you had a mutant for A, then you would be stuck at the white intermediate, and you would not be able to get yellow. And therefore, our two mutants must be E from the first pathway and B from the second pathway. And this implies that my dihybrid individual is going to be big E, little e, big B, little b, and when we do the dihybrid cross, we're going to cross this individual or self this individual to the same genotype, and that gives me my 4 phenotypic classes: big E over dash, big B over dash. So dominant phenotype for both the genes, or in this case you have enzyme for both of them. You could have dominant for the first one, so big E over dash, and homozygous for the recessive allele of the second one, or homozygous for the recessive allele of the first one, and dominant for the second one, B over dash. And lastly, we have our double mutant, which is homozygous for the recessive or mutant alleles of both the genes. And these 4 phenotypic classes are going to be present in a 9:3:3:1 ratio. Now we should work out the phenotypes of these phenotypic classes. So if you have one enzyme making allele of E and B, that means white will get converted to blue, and yellow will get converted to red. And in here we are assuming that you are homozygous for the wild type alleles of all the other genes. Therefore, you will get purple. In the second class, you have enzyme for the first gene, for E, so white gets converted to blue. However, you are homozygous for the mutant allele of B, and therefore yellow will not get converted to red. The yellow pigment will build up, and we will end up with yellow and blue, which the problem stated is green in color. In the third phenotypic class, we don't have the E enzyme, and so we do not get blue pigment and we are stuck at the white 1 intermediate, but we do have B enzyme, and so yellow gets converted into red, and therefore our phenotype will be red. And finally, in the last class, we are homozygous for the mutant alleles of both the E and the B genes, and therefore in the first pathway white does not get converted into blue, and so we are stuck at white 1, whereas in the second pathway yellow does not get converted into red, and therefore we are stuck at the yellow pigment stage. Therefore this individual will be yellow, and this confirms that our guess that it is E and B mutations that we are dealing with is correct, since we have recovered the 9 purple, 3 red, 3 green, and 1 yellow ratio. So the only thing remaining to be done, since this is the F2 generation and the dihybrids were the F1 generation, is determine what must be the genotypes of the pure breeding parents of the F1 dihybrid, and the pure breeding parents of the F1 dihybrid must be mutant for 1, homozygous for the mutant allele of 1, and homozygous for the wild type allele of the other gene. So big E, little b, and the other individual must be homozygous for the mutant allele of E and also homozygous for the wild type allele of B, and these parents would then give the F1 dihybrids, which would give us the modified ratios, phenotypic ratios that were posed to us in the problem. In the next part of the problem, we have a modified ratio of 9 purple is to 3 green is to 4 blue. The first thing to notice here is that the blue pigment is always there, since purple is blue plus red, green is blue plus yellow, and blue is blue plus white. Secondly, the presence of green suggests that there must be a mutation in B involved, as well as the presence of blue implies that there could be a mutation in A, so that if you're stuck at the white intermediate in the second pathway, you will result in all blue flower. So with this in mind, we can write down a potential dihybrid genotype, which would be big A, little a, and big B, little b, and this would be the self in the dihybrid cross. And this will result in our 4 phenotypic classes: dominant for A, dominant for B, dominant for A, but homozygous for the recessive or the mutant allele for B. And then you could be homozygous for the mutant allele for A and dominant for B. finally you could be a double mutant, homozygous for the mutant allele of A as well as B. And we can now mechanistically work through the pathway to determine the phenotypes here. So if you are big A over dash and big B over dash, that implies you have both enzyme to convert white to yellow as well as yellow to red, and your phenotype is going to be purple. And of course, we're assuming here that you are homozygous for the wild type alleles of all the other genes. If you are dominant for A, so big A over dash, that implies that you have A enzyme, and then you can convert white 2 intermediate into yellow. However, you are homozygous for the mutant allele of B, so you cannot convert yellow into red, and you will build up yellow pigment, and yellow plus blue gives green. So we'll have green here. Then in the third phenotypic class, you are mutant for the A gene, so homozygous for the little a allele, and you will be blocked here, and you cannot convert the white intermediate into yellow. Therefore, your phenotype is going to be blue. And in the last double mutant, you are homozygous for the mutant alleles of both the enzymes, and it doesn't matter because if you are a mutant for A, then you will remain stuck at the white intermediate, white 2 intermediate, and your phenotype is going to be blue. And here we see recessive epistasis, where A is epistatic to B, or in other words, the homozygous mutant genotype of A hides the phenotype, the mutant phenotype of B in the double mutant. And we have our modified ratios 9:3:3:1. But since blue, the last two phenotypic classes have blue color, we achieve a modified ratio of 9:3:4, which is what we were given. It's a situation with recessive epistasis. That means our F1 dihybrid is the correct genotype, and the only thing remaining to do is determine the genotypes of the parents. And in this case, in order to achieve this dihybrid, one parent must be homozygous because they're pure breeding for the wild type allele of A, and homozygous for the mutant allele of B, while the other parent would be homozygous for the mutant allele of A, and also homozygous for the wild type allele of B. And such parents would result in the dihybrid F1 progeny, who would then give rise to F2 progeny having the given phenotypic ratios. Problem 2 states, corn breeders obtain pure lines whose kernels turn orange or scarlet when exposed to sunlight. Normal kernels remain yellow in sunlight. A cross between the orange pure breeding line and the scarlet pure breeding line yielded all yellow progeny. Selfing the F1 individuals yielded the following ratio: 182 yellow is to 80 orange is to 58 scarlet. Explain the inheritance of this trait and use crosses to show how these ratios are produced. So first, we note that when we cross the orange line with the scarlet line, we get all yellow, that is all wild type. And that tells us that the orange and scarlet mutants complement each other and therefore must be mutations of different genes. Further, we note that the ratio 1:82 is to 80 is to 58. If you divide all the three numbers by 20, is roughly a 9:4:3 ratio. That is, it's a modified 9:3:1 ratio, and therefore the two genes must be unlinked. And so we can say that orange is the mutant allele, homozygous for the mutant allele little o, and homozygous for the wild type allele of the other gene, whereas the scarlet line is going to be homozygous for the wild type allele of the orange gene and homozygous for the mutant allele of the scarlet gene. And therefore, when you cross them, so we're going to do little o, S plus, cross to O plus, little s, we'll get an F1 dihybrid who is O plus over O and S plus over S. Now, the ratio 9:4:3 suggests that we are looking at recessive epistasis here, and in fact, orange, that is little o, is epistatic to scarlet, which is little s. Recalling the fact that epistasis means that in the double mutant, which is the genotype little o, little s, this is the double mutant, homozygous for the mutant alleles of both genes. The phenotype is of the gene that hides the other mutant. That means if orange is epistatic to scarlet, the double mutant will have a phenotype of orange. We should save this fact while we work out the dihybrid cross. So now we will carry out the dihybrid cross, and we will self these corn plants, the F1 dihybrids, so that we get our four phenotypic classes. We'll get O plus over dash, that's dominant for the orange trait, and S plus over dash, dominant for the wild type allele of the scarlet trait. Then the second phenotypic class is dominant for the orange, which is, and dominant is the wild type gene, so O plus over dash, but homozygous for the mutant allele of the scarlet trait. Or the third could be homozygous for the mutant allele of orange, and S plus over dash, so having the at least one dominant allele, the wild type allele of scarlet. And the last class is going to be the double mutant, which is homozygous for the mutant allele of orange as well as homozygous for the mutant allele of scarlet. And these are going to be present in a 9:3:3:1 ratio. And we can write down the phenotypes. Since the wild type is dominant, and we know that because of the complementation, when we cross the two mutant strains, all the progeny were wild type, and that tells us that wild type is dominant. So these are going to be yellow. Next, the next class is has the wild type allele for orange, and therefore it will not have the orange phenotype, but it is homozygous for the mutant allele of scarlet, and so this class will be scarlet. The third class is homozygous for the mutant allele of orange, and therefore these plants will be orange. And finally, we have the double mutant, which we inferred is going to have the orange phenotype, since orange is epistatic to scarlet, which we deduced from the 9:3:4 ratio that was given to us. And working out this cross confirms that we will get a 9:4:3 ratio of yellow is to orange is to scarlet, with the conclusion that the orange mutant is epistatic, must have been epistatic to scarlet.